Okay, so we are at coming towards the end of our symposium day. We have our last two graduate student scholarship awardees that would be presenting on their projects. First, we'll be hearing from Yue Feng, PhD candidate in Dr. Aaron Schimmer's lab, and then followed by Anna Novaleska, master's candidate in Dr. Clifford Lebrac's.
Okay, thanks for Sonia for introduction. So hi, everyone. Today, I would like to talk about my project of understanding mitochondrial protease ClpXP, its degradation machinery, and its function in acute myeloid leukemia. So ClpXP is actually a serine protease that's located in mitochondria, which will help maintain the protein homeostasis. And one of the reason why we're interested in this, because this protein is reported to be closely related to cancer. So ClpX is overexpressed in multiple malignancies, including AML, breast cancer, lung cancer, and etc. In particular, more than 45% of the primary AML patient sample actually overexpresses ClpX. And more like what Dr. Julia Pierre said, some of the cancer cells is still dependent on the oxidative phosphorylation. And when we're trying to inhibit or hyperactivate this protease, like chemically or genetically, we found that it will impair the oxidative phosphorylation function, and that has anti-cancer effect in AML. And lastly, also there are studies suggesting that the ClpXP would degrade the unfolded oxidative phosphorylation subunits in trying to maintain the respiratory chain function in mitochondria. So with this, we think that the ClpXP may be a potential therapeutic target in AML. And as for the complex itself, it is composed by the ClpX and ClpX. So ClpX is a chaperone that utilizes energy from ATP to unfold the protein, where ClpX is actually a serine protease that each monomer contains a serine, histidine, and aspartic acid catalytic triad. So under the inactive form, the ClpX would form into hexameric ring, while the ClpX would form into a heptameric ring. And in the presence of ClpX and ATP, they would form into an active complex where you have a tetradecamer of the ClpX, and on each end you capped with the ClpX ring. And by using the energy of ATP hydrolysis, ClpX would recruit the substrates and unfold their tertiary structure, and then fit the peptides into the proteolytic lumen formed by ClpX. So this is what we know about this protease, but what still remains to be elucidated is actually why ClpXP is degrading substrates and how it's achieving its substrate specificity. So interestingly, like in parallel with studies of human ClpXP, studies on bacterial homolog ClpCP found out that the ClpCP would recognize a phosphoarginine residue on the protein as degradation marker. So the heat shock damage protein in the bacteria will first be marked by the MCSBase kinase on the arginine residue, and then this phosphorylated arginine residue will be recognized by the ClpC, and the substrates will subsequently be degraded. So this led us to the question that whether human mitochondrial ClpXP would have a similar system of recognizing phosphorylated amino acids as degradation marker, and also how these markers would affect the protein functions in mitochondria in general. So to answer this question, firstly, we're just trying to assess how different individual phosphorylated amino acids would affect the ClpXP enzyme activity. So to do this, we did a cell-free enzyme activity assay where we incubated the tagged casing substrates with the recombinant ClpXP and also with increasing concentration of the phosphorylated amino acids. And here we actually record the fluorescence signal of the peptides as the enzyme activity. So as shown here in the figure, you can see on the y-axis is a percentage of the ClpXP enzyme activity, while on the x-axis is actually the concentration of the ligand. So with increasing concentration of phosphoserine and phosphothreonine, we see there's an inhibition of the ClpXP activity, while we don't see this for their dephosphorylated counterparts. And also to validate our data in the free amino acids, we also did the same experiments in the peptide context where we added each alanine residue on chain. So we have the alanine phosphorserine alanine and alanine phosphothreonine alanine peptides. We see the similar inhibition effect with even lower IC50 comparing to the free amino acids. So this is what we have for the cell-free enzyme activity data. Actually, other than this, we also conducted thermal shift binding assay, which is not shown here, but basically the takeaway is the ClpX would interact with the phosphorserine and phosphothreonine. So our cell-free data is actually suggesting that the serine and threonine phosphorylation may be important for the ClpXP substrate degradation. So we move on to the within the cellular system. So previous study have shown that the complex 2 subunit SDHA would interact with the ClpP, and also when we're trying to knock down ClpP in the AML cells, we see there's decreased activity for complex 2, and there's accumulation of damaged SDHA. So what I did is I first tried to confirm the interaction of ClpX, another subunit of the protease, their interaction with SDHA by using the proximity ligation assay. As you can see here in the top panel, showing in blue is the DAPI staining for nucleus, and the red is the PLA signal. So there is actually interaction between the SDHA and the ClpX. So with this and our cell-free data in mind, my next question would be how ClpXP would affect the SDHA serine phosphorylation level within the cells. So what I did is I did phosphoserine immunoprecipitation and then plot for SDHA. So on the y-axis shown here is the relative serine phosphorylated SDHA level in the cells, which is reflected by the densitometry. And on the x-axis, you can see we have different clones of ClpX and ClpP knockdown. So in both ClpX and ClpP knockdown cells in AML, we see there's increasing level of serine phosphorylated SDHA, which is consistent with our cell-free data that the serine phosphorylation may be important for the ClpXP degradation. So this is so far what we have for this project progress-wise. And now I want to switch gear and talking about how interdisciplinary aspect is involved in this project and how potentially we can engage each patient. So our project is actually a close collaboration among molecular biologists like myself, and also structural biology expertise and proteomics experts, and also chemists. So in collaboration with proteomic experts, we did the ClpX BioID, where we mapped the ClpX interactome. So this will provide us with a list of potential endogenous substrates for the protease ClpXP, and also provide insights on what are the potential function ClpXP is involved, and what are the pathways. And also in collaboration with the structural biologists, we're trying to solve the structure of the human ClpX and trying to visualize the binding sites for the phosphoserine. And if we're lucky enough, we will get the structure information, and this would provide information on the actual binding sites and facilitate the design of the phosphoserine mimicking small molecules that to inhibit the ClpX. So in terms of patient engagement, I think one of the key here is to try to communicate with the patient about the progress we have in the science field. So with affiliation with Princess Margaret Cancer Center, we have the chance to communicate with the leukemia patient and present them with the most cutting edge progress on the science field. So hopefully that will encourage them and make them more excited. And also our lab will participate in activities such as the Light the Night Walk, like founded by the Leukemia and Lymphoma Society, that with this event, we can walk with the Black cancer community and then hear from them to know what is the most urgent need with the leukemia patient and have that in mind when we're designing the future studies. So in summary, we found that the phosphoserine and phosphothreonine would bind to the ClpX and inhibit ClpXP enzyme activity in the cell-free system. And also within the cells, we found that ClpX would bind to the SDHA, and then the ClpXP protein level would affect the SDHA serine phosphorylation level in the acute myeloid leukemia. So moving on next, we were trying to visualize the phosphoserine binding sites on the ClpX by using the cryo-EM, and also trying to investigate the functional significance of the SDHA serine phosphorylation. And lastly, we can potentially develop the phosphoserine mimicking ClpX small molecule inhibitors. So with that, I would like to thank my supervisor, Dr. Aaron Schimmer, and all the support from my lab members, as well as our collaborators from Dr. Brian Brown's lab and Dr. Mohammed Jafari's lab. And lastly, I would like to thank MITO2i for provide this opportunity for me to present here and also financially support this project. And that's all. Thank you.